Good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Eltem Technologies, welcome you all for joining us today in the last but not the least session of our Women's Day special webinar series. We are excited to have you all here. Today is 8th March, and we all know this day is celebrated as International Women's Day. So I would like to wish all the women as a happy Women's Day. If one can portray a woman, she is the epitome of strength, courage, power, determination, and of course, dedication. It's a day to express our gratitude to the women in one's life, whether it is professionally and personally. These women have a fierce and the robust spirit which makes them excel in their respective areas. Altum Technologies salutes all those women out there who are just doing wonders in their lives and around the globes. And of course, how can I forget to wish you all a very happy Holi? May God paint the canvas of your life with vibrant colors. It won't be complete without expressing my gratitude to each of you for contributing to the success of this Women's Day special webinar series campaign. I always believe in growth is never by mere chance. It is result of people working together. We will be conducting a quiz. That quiz link will be shared in the chat box, and lucky ones will have a chance to win the gift voucher. So let's start with our today's session, which is on next generation sequencing and its application in agriculture. Bioinformatics is already, begin to demonstrate how it can significantly influence worldwide agriculture research and the development. We all know how rapidly NGS technologies have advanced in recent years, and this has led to the wealth of insightful discoveries. Single nucleotide polymorphism, which is called as NIPS, is the most abundant DNA sequence variation present in the plant genome. Sorry. NGS technologies bring new tools and concepts that can enhance the preciseness and efficiency of plant breeding, such as cost effective, high throughput genotypes technologies for the sustainable agriculture and crop improvement. Our speaker of the day will be elaborating on how these techniques will be applicable in the agriculture. So it's time to introduce our speaker of honor, Dr. Nisha Sharma, presently working as an assistant professor, Department of Bioinformatics at the Gujarat Biotechnology University, Gandhinagar, Gujarat. Dr. Singh holds MSc in Bioinformatics from Bhubaneswar, PhD in Biotechnology and Bioinformatics from the National Institute for Plant Biotechnology, New Delhi, and postdoctoral training from the Institute of Genomic Diversity, Cornell University, Ithaca, New York, USA. Dr. Singh served as DST Inspire faculty at National Institute of Plant and Biotechnology, New Delhi. Her research interest lies in genomics, proteomics, and bioinformatics. She is especially interested in developing cost effective, high throughput genotyping technologies for genetics and molecular breeding application in the diverse species. During her PhD, she has developed the first patented Gene 15K SNP genotypic chip as well for genetic and the molecular breeding application in the rice. Dr. Singh has developed the bird's first halotype map of tea to facilitate the understanding of tea genome evaluations and tea metabolite pathways. Dr. Singh has developed several genomics and bioinformatic tools and pipelines such as heterozygous genome assembly, gene based halotype pipelines, SNPs assay development tools. Dr. Singh worked in several international genome sequencing projects, including wild rice, wheat, mango, jute, pigeon pea, and the tea. During her research career, she got many scholarships and awards. She has published more than 59 international research publications in the reputed journals and 3 patents, and of course, 2 books chapters. She has more than 1,600 citations to her credential. Dr. Singh has served as a reviewer for several prestigious international journals. She gained several prestigious awards as well. So, without further ado, let's start the session. Over to you, ma'am.
Thank you so much, Sakshi, and wish you a very happy Holi and happy International Women's Day. And thank you so much for providing this opportunity so that I can share my work, what I have done throughout till using this platform. So thank you so much and for the nice introduction. So should I start now?
Yes, ma'am. Please.
Okay, so my slides are visible now.
Yes, ma'am.
So yeah, today I'm gonna to talk about the next generation sequencing technology and its application in agriculture. Right. So currently I'm working as an assistant professor at GBU, Gujarat Biotechnology University, which is newly established university dedicated to biotechnology only. So let's start with the genomics. What is genomics? So we know that the genome was a term coined by the Hans Brinker in 1920 to define the complete set of genetic information present in an organism, and the branch of study of genome complete structure, including its structure and function, is known as genomics. So the genomics was coined by the Thomas Roderick in 1986. If we talk about the types of genomes, so there are broadly two types of genomes. One is prokaryotic, the other one is eukaryotic. And in eukaryotic, we are having plant genomes and the human and animal genomes. So the plant genomes considered to be more complicated than the human and animal genome, because the plant genomes we are having additional genome, that is chloroplast. And in eukaryotic, mostly we are having nuclear genome, mitochondrial genome, and in plants we are having the additional chloroplast genome. So that's how it's make more complicated. So I'm working on the plant genome, so it's pretty complicated because we are dealing with this different types of ploidy. Yeah, so the subfields of genomics, it comprises structural genomics, comparative genomics, mutational genomics, and functional genomics. So my research areas lies in and across all the four different domains of genomics. So I deals with all the structural aspect, comparative genomics like synteny analysis and evolutionary analysis, mutational like SNP based assay design and their application, and of course the functional validation of those identified candidate genes. So what are the goals of genomics? So let's say if we talk about the earlies, where we are having only the Sanger sequencing, so it took longer time to develop any gold standard genome sequencing project. Let's say we can see that the in case of human genome, also the first model plant Arabidopsis, so it took longer time, but now the advancement of this NGS platforms, and we are having several different now we can able to chase this goal very quickly. So the genome sequencing we are having, first we either we can go with the guided or de novo, and after genome sequencing we have to perform the assembly, because the sequencing are in the small stretches like context that we need to make the scaffolds, and that whole process is known as the assembling of the genome sequencing. And after assembling we have to provide the annotations, like what are these particular like scaffolds means, like what are these ATGC comprises in. So we need to have the annotations, like what are the functional function of that particular stretch. So for that we perform the annotation, through that we know the exact function of that particular gene. And since we are having lot of use data of all these sequencing project, we are generating plethora of data. So where we will store this, so we have to make the genome database. So since the genome sequencing project launched, so the next step after gathering all the information, we have to create the databases where we can have all the information about the genome sequencing, assembly, annotation, SNPs, variants, and all. And after database development we can identify the genes of our purpose where we want to target, and perform some sort of genome editing, or some functional analysis. And of course during the genome sequencing process we can identify the variants in terms of SSR, ESTs, and SNPs. SNPs are the most abundant DNA marker in the plant genome or any genome, and they are mostly biallelic and occur at high density in the genome, right? So that's why they are people now using this for the personalized medicine and sort of personalized, because every individual is 99.9% similar. 0.1% variation is occurred due to presence of SNPs. And then using this SNPs, identifying these SNPs through different approaches like GWAS and the other approaches which I will discuss later, we can go for the gene and genome editing to develop superior crop varieties. So if we talk about the plant genome sequencing approaches, so the very first, in case of plant genome, we are having the Arabidopsis thaliana. The genome was sequenced in year 2000, and it is having 125 MB genome size, comprises total 25,000 genes. And later 5 year, we are having gold standard rice genome sequencing, which is released in 2005, comprising 36,000 genes with 389 MB genome size. And this the rice genome sequencing is considered to be one of the gold standard genome sequencing project, and there 13 countries are involved, and India was also the part of this particular genome sequencing. This was the first time where the our Indian scientists have took the initiative to do this genome sequencing project, and they fight a lot for this, because I was blessed to have that a chance to meet and work with that such great scientist at NIPB, National Institute of Plant Biotechnology, where they have allotted the short arm of the chromosome 11. So India was the part of the rice genome sequencing project, where we sequenced the short arm of chromosome 11. And then after this, our lab was quite stabilized in this area, and now we do a lot of genome sequencing of other crops, like tomato, wheat, pigeon pea. But the pigeon pea, why I have emphasized here, because it's the first legume genome who's decoded fully in India by Indian scientists. It's a complete Indian initiative, and its genome size is 852 MB, and total 47,000 protein coding genes are present. Now we can do sequencing, and now we are having more than 300 plant genome sequence in the databases. So these all about the sequencing. Similarly, with the number of genome sequencing right now is accumulated, because due to the our technology advancement. So initially we have to do with the first generation sequencing, like the Sanger sequencing was the prime source for doing the sequencing, and then later on we have to perform the second, we can use the second generation sequencing, and now we are relying more on the third generation sequencing, like PacBio, Oxford Nanopore, Mini Ion, Promethean, sort of things which are having long reads, and now we can have the chromosome level of assembly at that. So the next NGS is the revolution in arena of genomics, of course, and it's a great accomplishment, because the sequencing is getting cheaper day by day. Because of this advantage, we can able to create the 3000 resequencing project of rice, which was developed by the ERI. But everything brings some new challenges. So similarly, in case of NGS, we are having the new omics emerges, that is computomics, because since the sequencing cost is depleting, but our computational cost is increasing because of this big data. We are having greater number of yields in terms of the sequencing data, but how to analyze and how we can store this data, this is the main challenging area in this. So that we need to address. So that's why we need to more focus on the bioinformatics pipeline to develop the more stringent and strategical, so that it will reduce the computational cost and at the same time enhance the capacity. So the sequencing platform I have mentioned earlier, that there are two major sequencing technology: short read and long read. In the short read, we use Illumina, and which has yield of 150 base pair with 0.1% error rate. In the long read, we are having PacBio and Oxford Nanopore. Here we are getting the high number of yields, but the error rate is 10%. But what I feel personally during my research, that the error is not because of the technology. Error is due to we don't have the great this pipeline and the software available to address and to tackle such a huge volume of data. So the scientists, especially the bioinformatics people, needs to take the lead in this direction, and to develop some of these tools, so that it can easily handle these are the steps in the genome assembly. Like first we are having the raw sequence read, and then we have to assemble these reads in the form the contigs, and then after that we can do the polishing and scaffolding, and we fill the gap with the help of N, and then finally we can do the pseudo molecule or chromosome level of finishing. So there are the two assembly strategy. Initially we used to have the Dubridge graph methods where it's based on the Kamer, the values. And now we are having, since we are having the long read sequencing, so it's based on the overlap layout consensus, right? So yes, big challenges is NGS is the data analysis and implementation, because we are having lack of really very good analytics person, especially in the bioinformatics, because since it's an emerging field and we are having not so much of expertise, especially in India, to do such kind of analysis and all those. So we have to rely on others. So that's how I encourage. It's a very booming area, so we have to take up a lead and solve these challenges so that we can play around with this big data science and big data analytics. So my research is focused on that. So now I come to the how these NGS and genomics can help in the crop improvement, and I have addressed all these applications using this big data analytics and NGS. So first we can go one by one. The first one is the harnessing genetic diversity. So we can analyze the genetic diversity within the species and across the species. Within the species we can have the several genotype where we can perform the pangenome sequencing and also identify the different types of genes, domestication related genes, agronomically important genes, and identifying those genes we can transfer to the our elite cultivated variety, so that can we have a superior breeds. The second one is across the species, what are the variation present across the species and how we can use those genes to mitigate the stress, both biotic and abiotic level of stress to the other crop species, how we can transfer these information to the other species. So that's how we can map the different variants across the traits, same in the case of human also, and also in the plants. For instances, here you can see the image of semi-dwarf rice variety, and this initially why this happened, because of the SD1 gene, semi-dwarf 1 gene. These are the genes which are responsible for the dwarfism in plant. So loss of function of gene and gain of function of genes. If the gene is absent, then the and then the plant will be taller, and if the gene is SD1 gene is present, then it will be the shorter. So that's the thing. The third one is the population stratification. We can have the collection of diverse genotype, and we can identify their pure breed and also the admixture population within the different subpopulation group. For instances, here we have collected more than 300 Indian wildlife accession from different ecogeographical region of India, and we try to stratify based on the SNP marker, and that to the single copy gene, because they are evolutionary conserved genes, and identify that the what are the gene response, what are the variety which are purely wild type, what are the admixture types and cultivated types. So we can see that most of them are the mid gangetic, which is the completely wild type, are the pure type. And these here you can see that the some of the pro aus progenitor of aus type is also the pure type, and progenitor of indica are the less pure, and most of them are the admixture. And this admixture we can estimate with the level of fixation index within the population. And what is the marginal effect of changing one allele to another allele? So here we can see the evolution in population genetics with the help of like kinship matrix and adaptation and their mating system. Also with a single mutation, how one genome leads to change and make the another genome. So that's why it is really essential to do not only one gold standard genome sequencing, we need to perform the pan genome sequencing, so that we have a representative genes present across the different genotypes. And what are the different physiological conditions like G by E impact we can study, because initially people are ignoring the environmental impact. Now they are taking into consideration, and which is really very key, because some of the genotypes of plants may survive here in the Gujarat but may not in the other part of the country because of their environmental and some other favorable conditions. So that's thing we need to check with the multi location trials and all. And the fifth one is the trace the evolutionary history of the origin and domestication of Indian wild rice and their relationship with the cultivated rice. Our war with China is not only restricted to the boundaries, but it's beyond the boundary, because Chinese people claim that the rice did originated in China, not in India. But the theory is not completely correct, because rice has a polyphyletic origin, not the monophyletic origin. And based on the some key agronomically important genes that I have proved that the wild type of the Indian wild types are originated in India, not in China, because they are the people use, they are more of the sticky kind of rice, but here we use diverse set of rice. And most of the waxy genes are absent in the our indica rice variety, but it's present in the japonica variety. And they are having different sort of wild type. We are mostly blessed with the Oryza rufipogon and Oryza nevada. And also we can develop the species specific marker, so that we can map the important traits and that. And further we can use the high throughput phenotyping and genotyping, and we can perform the genome wide association analysis and identify the associated loci and SNPs which are responsible for that particular trait. And we can further validate the functional analysis and understand the molecular mechanism and go for the pathway analysis. And now the genome editing using Cas9 and other system, and we can we can generate a superior breed. So this is one of our study where we performed the genome wide association analysis for the anaerobic germination of deep water rice variety of Assam population. And you can see that this study is morely on the Baudhan, and we have identified the two key genes which is responsible for the internode elongation in rice, and which has a kind of escaping mechanism when the floods come, and so that rice can have the internode elongation and it survive in the flooding condition, because Assam is the most flood prone area. So these are the two key genes we have identified the GWAS study, that OSXDH1 and SSXT family protein, which is responsible for internode long elongation. So if we can identify this gene and transfer this particular gene to the other susceptible variety, so that it can also have that particular trait, and we can use for the this variety. Then the most recent application in the breeding aspect of the crop improvement is the gene based haplotype estimation. Now we are using this for the haplotype based breeding concept. So it basically assists the breeders to decides like which of the genotypes they have to plot in that particular area. So it's really very great achievements, so that you cannot have to plot you cannot have to take all the varieties. Just you can make one mini core based on the haplotype analysis, and you can uh, 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 provide that particular seed sample to the farmers, so that they can uh, grow that particular variety in their fields. And of course we can go for the varietal identification seed purity testings. And most importantly, now the concept is gene to plate. Like initially one gene, one a particular study. Now gene to genome, we are dealing with the genome. Now genome to the field, so that what is the impact of genome in the field, we are just taking in the thing. And now we are into the translational aspect, like the plate, what's really coming in the fields, what we are getting really. So it's all depend upon the consumer preference. So that's how we will edit the things. Similarly in case of this, like genome editing, we could able to do that, the bacterial blight resistance, fragrance rice variety, high grain quality, and large grain size in case of rice. Similarly in case of wheat, we are able to develop the powdery mildew resistance, high amylopectin content. We are able to edit like and make the tomato purple and powdery mildew resistance. We could able to enhance the high oleic and low linoleic content in the soybean. And yes, we could able to escape against the citrus canker disease. So these are the few wonderful examples how we can enhance the crop by genome editing techniques. So of course the genome and NGS have certain challenges, because some of the genomes are really very complex. If I talk about the wheat genome, it's a 16 gig, and we are having the high level of heterozygosity, degree of repeatedness. In case of maize, it's highly repetitive. So and lot of jumping genes is present. So dealing with these repetitive element is really very hard to get to study its genomics. And yes, of course, genome assembly algorithm which designed primarily for the human and mammals, but not for the plants. So the plant scientists really need to think very rigorously and develop some of the good tools and software, so that the plant genomes can tackle. And of course some of the they have just simply released the rough and draft variety of the genome sequencing, which doesn't make more sense, because if you want to really do deep genetics and genomics, the quality of genome should be gold standard. Lack of expertise in the field, and of course the funding agencies is now they are saying that why genome sequencing, what's the need. But yes, genome sequencing is really needed, because you can see that I talk about the example of human genome, so it's recently finished, because earlier it has gap, but now the human genome is gapless. So similarly, in the crops, there are a number of crops which we have released the draft, but it's not yet gold standard. So that's why we need to more funding, so that we can able to have a really great genome sequencing, so that we can address and study its more deep genetics and genomics and enhance its crop improvement. Because our India, especially few of the variety which is really important, and like horticultural fruit and crops, they are really important for the India. For instance is mango, pigeon pea. These are the crops which are really we need to consider for the India, because why Western world will invest, because they are more relying on the wheat or maize. So that's how. So now I come to the my research work. So we have developed the first unique genic 50K SNP genotyping chip for genetic studies and molecular breeding application in rice. Why I could able to do that? Because we are having the gold standardized genome sequencing available, and so that using those sequencing information, we can have able to develop the this platform, the SNP genotyping platform, so that it can accelerate the breeding application in rice, and also the genomic selection, and we can able to reduce the breeding time basically. So that's the key thing in this study. So the SNP genotyping platforms are the two type. One is the low multiplexing with high sample throughput, another one is high multiplexing with low sample throughput, and both the platforms have varied application. For instances, low multiplexing with high sample throughput, we are having population structures analysis, we can perform foreground selection, diagnostic purpose we can use, and seed quality control. Similarly, high multiplexing with low sample throughput we can use for the evolutionary analysis, genome-wide association mapping, background selection analysis, genomic selection, and diversity analysis. And this is the complete pipeline how we have able to develop the 50K SNP genotyping chip. And this is the unique chip, and we have patent for this chip, and gain lot of attention in the rice researcher, and they have used, and we have good number of citation of this particular work, because this is the completely genic chip, and we have taken into account of the single copy genes. So it's a thumb rule that in any of the genome, it's around 10 to 15% of genes are the single copy genes, right? So we have taken four different category of genes here. The first category is conserved single copy gene common in wheat and rice. Second category is single copy gene unique to rice only. Third category is agronomically important clone rice gene, and fourth category is multi copy rice genes, where we have kept as a control, identifying SNPs using different pipeline, and we have integrated and make our own in-house pipeline, and the mine SNPs across four different category of gene. And for the FE designing, we have to take into account of 35 bases of sequence from either side of the SNPs at 36 base position SNP lies. So the whole stretch of ATGC will be 71 base pair. So the 71 base pair array design, and at 35 bases from either side of the SNP. So the 36 base position SNP slice. And after all the quality check and all the p convert scores check, we finally synthesize the SNPs with the 50,051 high quality SNP marker. And then the final version of chip is like this. And at a time we can genotype 96 sample, and in a single well this 50,000 marker is embedded. So you can see the data point like 50,000 into 96. So this much data we are getting with from the one chip. And this we have run more than I would say like more than 12 chip we have run for the rice. So you can see you can imagine the data point we have, and then we use this data for the varied application and validation purpose. So percentage of gene, if I talk about, so this highest percentage is coming from the single copy gene unique to rice only. These are the gene which is unique to rice only, so that is 79%, followed by 90% coming from the conserved single copy common in wheat and rice. So this particular set we can use for the wheat purpose also, wheat breeding program also, and 1% each from the multi copy gene and agronomically important rice genes. So the interesting fact you can see here that with the 79% we are having only 44% of this SNPs. Why? Because these are the more recent genes. But with 19 percentage of genes, we are having total 52 percentage of SNPs. So why we are having this? Because these are the more ancient genes. So we are having threefold higher number of SNPs here. So getting so because over the period of time more point mutation accumulated, and so that we are having more number of SNPs here. Similarly, in case of AGCR, we are having 1 percentage of gene with 3 percentage of SNPs. So it's again why? Because most of the agronomically important genes are larger in size, so that we can able to mine more number of SNPs. And the percentage of SNPs in different reasons. So the 41% is coming from the intron, and 38% from both your synonymous and non-synonymous reason, 14% from 3' UTR, and 7% from the 5' UTR reason. And this is the physical map of the rice, and rice has total 12 chromosomes, and you can see that the density, how they are equally distributed, and they are quite dense. And on an average around on an average around per gene we are having 6 to 7 SNPs. And the distance between adjacent SNP here is less than 1 kb, so they are quite dense. And you can see the chromosome wise distribution of number of SNPs. So the highest percentage is coming from the chromosome 1, followed by 3 and 2. And chromosome 11 is mostly the disease resistance genes we are having, and it's mostly in the most of the crops. Chromosome 10, 11 and 12, these three chromosomes are the disease resistance genes. We can find more number of disease resistance genes in in these three chromosomes, right? And these are the these are the some of the pictures of the collection sites where we have collected Indian wild rice accession. Now we are having more than 1000 Indian wild rice accession from different ecogeographical region of India. Like especially we have targeted the 9 ecogeographical regions of the India, and we have made one beautiful databases of Indian wild rice database, and where all the morphophysiological information along with the marker information we have here, and so that we can go further in the GWAS or other sort of analysis. And it's a great platform and great genomic resources for the rice breeders and the scientists. And this is the paper we have published from the development of the single copy 50K SNP genotyping chip. And because of this chip we are able to deduce that the rice did originated in India, especially the Indian wild rice, and not in the China. And we have the patent of this, and we got this appreciation letter from the ICAR, DGICAR. And if I talk about the application of the 50K rice chip, so it's used in the haplotype based phylogenetic analysis. And we have here this study is based on 128 diverse rice genotype, and they were beautifully stratified into four different group. The group 1 is entirely wild type, group 2 is the aus type, group 3 is the indica type, and group 4 is the japonica type. So here you can see that with the this with the help of this 50K SNP genotyping chip, there is a clear cut distinction between the indica type and japonica type. And the other application, which is the completely breeding application, where we can see that the able to deduce the background genome recovery from the recipient parents, right? So there are the four mega rice varieties: Swarna Savan, Samba Masuri Savan, Savitri Savan, and IR64 Savan. So with the help of this 50K SNP genotyping chip, we can able to analyze the parentage recovery from their recipient parents. So the highest genome recovery we can see in case of Swarna Savan, that is 97.7%, followed by Savitri Savan and IR64 Savan. But the Samba Masuri Savan is quite less parentage recovery from their recipient parent, that is 78.7%. So because of this chip we are able to deduce that that this particular genotype require more background recovery from the recipient parent, which rice breeders can take several years to able to get this information. So this is the basic, this is the one of the great application of the 50K rice SNP genotyping chip. So it's basically reduce the breeding time, right? And the because most of the conventional plant breeding method based on the rigorous phenotypic selection, and it's all depend upon the breeder experience and their eyes. So if we able to deduce this information and find figuring out in the molecular level, so it save our time or also the cost and lots of manpower and labor. So let's say if one variety took almost 13 to 15 years to release, so because of this type of genotyping technology we can able to release one variety within 3 to 5 years. So see, 10 years we can able to save. So that's a great contribution. So apart from that, we can able to perform like phylogenetic analysis, QTL mapping, evolutionary study, background analysis, genotyping, and of course biomarkers for the drug discovery and other purposes. So the property of this 50K SNP genotyping chip, this is the single copy gene based SNP chip design, and it's non-redundant high quality SNP probe. It has a high sample success rate, 99.7%. It has high assay reproducibility and high genotyping call rate, and on an average around 6 to 7 SNPs present per gene. It has less chance of cross hybridization. It's quite cost effective because at a time we can genotype 96 sample. So it's like automated and multi-purposes also. And then single chip you can design like the multi-species format also, like in a one one fraction you can have rice snapes, the other pigeon pea, chickpea, and maize, like that. It's also available in the multi-species format. So these are the novelty and significance of this particular work, so that with the help of this 50K SNP genotyping chip, we could able to characterize the Indian wildlife germplasm, genetic background analysis, association mapping, QTL mapping of important agronomically important trait, and we can use to identify the novel haplotype present in the genome. We can estimate the genetic diversity, QTL mapping of the salt tolerant and bulk segregant analysis. We can study the evolution and domestication of the rice, and two submergent tolerance rice varieties, Ranjit Savan and Bahaduran Savan. They are the submergence tolerance variety, has been able to release with the help of this 50K SNP genotyping chip. And most important application, it helps in the reduction in breeding time. Now I come to my other research work. So apart from that, I have also developed the some of the low density SNP genotyping assays for the Illumina platform, and they are OPAs, oligopool assays. So this is basically for the characterization of Indian wildlife germplasm, and you can say that for the population structure analysis we need genome wide coverage for the unlinked marker, and mostly the low density marker is ideally considered for the population structure analysis. So here what we have selected, what are the basis of selection of the marker. Actually we try to cover the entire length of the chromosome, so the two from the top and the two from the bottom of the chromosome, so that it could able to the complete the entire length of the chromosome. So here, and the rice we are having total 12 chromosomes, so we have taken four snips from each chromosome, so that it has the full coverage. And using this 48 plex, we could able to estimate the population structure analysis and genetic diversity of the newly collected Indian wildlife accession. And this is the phylogenetic analysis of total 418 Indian wildlife accession: Oryza sativa, and Oryza rufipogon, Oryza nivara, Ows type, and Indica type. And also we could able to study the origin and domestication of Indian wildlife and their relationship with the cultivated rice. So as I mentioned, that there are the two hypotheses on rice origin. One states that rice has monophyletic origin, the other one is polyphyletic origin. Monophyletic origin states that a single origin model, Indica and Japonica, were domesticated in the same region from the wild ancestor, whereas the polyphyletic origin stated that the multiple independent domestication, both Indica and Japonica were domesticated separately and in different geographical part. So this was the like 2 decade back. People are more claiming toward this, because no one is like bothered about like why we should bother about the origin. What does it matter? Anyway, we are getting food, so that's fine. But now people start realizing, no, otherwise everything was later on we can think that's coming all from the China. So right. And so then people started doing this some of the this study based on the some key agronomically important genes, and also like and, some of the fossils record also, they have traced back and identified that the rice has the polyphyletic origin. And most recently this is the widely accepted both from both like from India also, and also one of the UK group, they have also published, like Kim et al, they have also supported our hypothesis. So based on like 13 key agronomically important genes, we try to address this particular question. So let's say RC gene, which is responsible for the pericarp color. So the modern rice variety we are having white color pericarp, but ancient wild type are having red color pericarp, green color pericarp, and also black color pericarp. Similarly with the hull color, grain size, and also amylose content, amylopectin content. So these are the some key important genes which can tell us like how rice has originated, and what are the pattern of domestication, and how they have gained some of the genes and lost some of the gene over the period of time. So this is the you can see that the morphological variability in Indian ORSE or Oriza rice wild rice succession. So first one is the auricle color. So you can see these are the auricle colors, and the second one is internode pigmentation. So see, internode pigmentation is varying across the wild type, and seed coat color. You can see seed coat color is also varying a lot. Extent of awning, because the wild type are having long awns, but the cultivated having short awns. On colors, wild type having red color awns, but the cultivated one having the green color awns. Panicle type, the cultivated is quite compact, but the wild type is spreaded one. So these are the some morphological phenotypic features which we can easily distinguish from our. But what are the molecular mechanism behind it? So we try to address this with the help of this haplotype based network analysis. And you can see that based on the RC gene, which is responsible for the pericarp color. So all the red one is the wild type. So the wild type are all are having this red or brown color pericarp color, and ours type is having green and white color pericarp, because the RC gene is start losing their function. And then the modern recent variety, which is all nested in the haplotype 1, which is comprising this our japonica type, indica type, and aromatic type. So you can see the variation in the green length, but the pericarp color is white. So you can see how the molecular level they are correlated. So that's how we try to address this. Similarly, we are able to do with the GS, grain size genes, also with the haplotype based phylogenetic analysis in GBSs. So GBSs we find very great structure, that all the haplotype H2 is nested, wild type is nested in the haplotype 2, with the small fraction of this south type. And there is lot of variation we can see in the case of japonica, the purple one. But the all wilds are conserved. So we can say that the GBSS gene, the japonica variety has a lot of variation of this GBSS gene, the amylose content. And but in indica type, we are having the same patterns of GBSS genes, and they are like, so that's how we can easily distinguish how indica and japonica were bifurcated. Similarly, the other genes we have tried to analyze and address. So I'm just keeping this in summary. I would like to conclude that here of this study, that Aus and Indica group of rice cultivated were domesticated in Indian subcontinent, where they have their closest wild relatives such as Oryza rufipogon and Oryza nivara. And results supported that rice has a polyphyletic origin, and mid Gangetic plain, upper Gangetic plain, and eastern hill region of the India are the source of Indian rice gene pool. And diversity within this wild rice is of immense value for rice improvement, especially for the impending climatic conditions. And we can utilize this wild rice resources, and we can meet the our projected demand of this rising population, because it is estimated that by 2050 it will be reaching around 10 billion. So to meet the projected demand of this rising population, we need a sustainable agriculture system. In this context, these wild resources play a key role, so that we have a climate resilient or rice variety or crop variety, so that it can tackle all the biotic and abiotic stresses and survive. And now come to the whole genome wide association analysis. So I performed this GWAS study in the this deep water rice of Assam, and we have registered two germplasm from this study, now the Nigeri Bawandhan and also the Sagrafala 2. And apart from that, I was also working on the genomics and proteomics resource and tool development in different crop species such as wheat, mango, cassava, pigeon pea, and tea. And currently we are working on the mango genome sequencing project. And mango genome sequencing, we are trying to sequence about the amrapali genome. And amrapali is nothing, it's just a blend of neelam and dasheri. So, and India is the mother of mango. So that's how the Indian scientists need to work on this. And also it was estimated by the fossils record that the Magnifiera indica has been originated in Damalgiri Hill of the Meghalaya, right? So its estimated genome size is 439 MB, but the major issue with this particular crop is its heterozygosity, because because in the mango we can able to see lot of heterozygosity. So that's the key issue. That's why we need a very good genome sequence information to work on this particular crop for its improvement. And also it's a like horticultural tea crop, so that's we need to take care. So mango is considered as a king of fruit, and this we have more than 384 diverse mango varieties like Pusa, Kesar, Alfonso, Neelam, Deshari, and we perform all the genotyping of this. But we are having tri-spinning assembly of both Neelam, Deshari, and Amrapali. So in with this study we can able to identify these are the this is the comparative genomics where we can able to identify the mango is comprising total 46,395 genes. Out of this 18,141 genes is unique to mango only, because these are the set of genes which makes mango, right? And the other genes are the two-way diagrams which shows that for instance in citrus 11,349 genes are common between mango and citrus, and citrus comprises total 29,655 genes. Similarly with the pineapple, watermelon, apple, banana, like that way. But with this study citrus is quite similar to the mango. But recently we have identified that the lychee is sharing more synteny with the mango. And similarly we have fabricated 80K genic SNP genotyping chip in the mango. And here we have sequenced total 24 diverse mango variety. And we are having five different kind of categories. One is the clone horticulturally important mango gene. The other one is disease resistance defense response like mango gene, single copy mango gene, and conserve single copy gene between citrus and mango, because we found that they are quite similar and they share more synteny as compared to the other crop. So finally the chip was synthesized with the 80,816 high quality SNPs probe. And the application of this 80K chip we use for the genome wide association analysis, the agronomically important and pomologically important traits, and we identified very important and key SNPs and genes responsible for the carotenoid and fruit length, fruit weight, polyembryonic, alternate bearing, because alternate bearing is really very interesting character in mango, because sometimes a mango bear fruit alternate years. So how what's the mechanism? So all these we try to deduce with the help of this. Similarly, I performed in collaboration with the University of Bristol this population structure and genome wide association analysis of phosphorus sufficiency trait using 35K wheat breed Fe arrays. And we try to estimate what are the effect of P plus and P minus and the wheat diverse wheat genotypes. And here we have taken more than 369 diverse wheat lines. And wheat is the complex genome, and its ploidy is also like hexaploid, tetraploid. So this is the tetraploid wheat. And we try to identify some of the important gene and perform the analysis. And this is the study I performed and developed the complete pipeline. And this is the first report from the India, the high and the high and deep haplotype map, hap map of T, where we can able to identify and make the complete comprehensive pipeline for how to mine the variants, true variants actually, so that it can further utilize for the genomics and improvement of the tea plant, because tea is equally important for India and it's considered as cash crop. And we have also make one wonderful database where we can perform all sorts of bioinformatics analysis on the tea plant. And now this is my project funded by the Department of Science and Technology, Government of India, DST Inspire grant, where which I'm working on Arhar. It's a pigeon pea. So we have developed 62K genic SNP genotyping chip of Arhar. And this 60K chip, you can see that we have published it, and we try to identify the population structure, because also Arhar is not the pigeon pea is not being mm, uh, is kind of an often an underutilized crop, because only the Indian people and some of the Myanmar of South African countries, they utilize this particular crop, but still they lack of the funding. So the Indian scientists, we took the initiative to develop the good genomic resources, and also try to identify some of the key important genes, and also the some of the high protein lines, so that we can contribute to the farmers, and so that per hectare we have the high protein gain in terms of. So we try to perform some of the robotics and drone for the fields for the high throughput phenotyping, because for the high throughput genotyping, we need to have the precise high throughput phenotyping. So we try to some map some of the trait manually, but most of the traits like plant height and all those things, leaf shape and leaf angle, try to map with the robots. So this is the glimpse of my work of DST. Like we have developed this 62K chip, and then phenotyping of these important traits like seed coat color, because it's directly related to the your antioxidant activity, and also the your biotic stress, seed weights, total seed total protein content, STPC, amino acid estimation, and resistance starch. Well, resistance starch is very less in this particular thing, but my main focus is on the total protein content. And mine by measuring all these traits, and also apart from that, we have mapped the pod length and number of pod color, flower colors, and leaf shape, leaf angle, plant type, plant architecture, all those things we have mapped, and performed the genotyping of those screened phenotype, and performed the genome wide association analysis, and identify the SNPs. And the study is still underway. So we have mined few of the traits which are really important, and simultaneously we develop one of the PGNP omics database, where all the uh, multi-omics information is available, starting from the phenomics, genomics, transcriptomics, proteomics, and and all the genotyping information in this. And we have screened the high protein lines. So these are the high protein lines. Why PGNP is mostly known for the maximum ranges comprising 16 to 22%, but in my study we have screened more than 800 different lines, and also made the one mini core collection of almost 400, and out of 400 we could able to identify there are the few varieties, or few different genotypes collected from different countries, also the collection of different countries, and also the land races and mini core collections are having protein content more than 25%, and few like 10 to 12 we are having more than 30% total protein content. So they are really great resources, where if we talk about in terms of protein yield, so it will if we plant that particular variety, so it will give you net protein gain in higher amount as compared to the other one. And also it can address the our sustainable goal development second, and it can address the malnourishment issues which is prevalent, most prevalent in the developing countries like us, India and South Africa. So my work is toward that. And so we try to map the qualitative traits like flower color, flower streak patterns, sheet coat color, and different, because these all are the traits which are interlinked, and also the quantitative traits which we can able to measure. So you can see in this picture, like we are having total 8 or 9 the spot, like the seeds, but in a few of them you can have only 3 or 4. So how these patterns, so we have seen lot of genetic diversity in this particular plant, and perform the genome wide analysis, where I'm showing you few of my results. And we perform the GWAS on 282 pigeon pea varieties, and we find some of the significant SNPs. Further we need go for the validation. And after identifying high protein lines, we try to make F1 with the high protein line with the intermediate type, so that if we can able to gain 1 or 2% of protein in this recipient one, so it will be also a great contributions. And we developed the high density linkage map based on some important traits like this primary branch per plant, pod bearing length, and pod bearing length, primary branch, and number of seed per pod. So these are the important traits. And also we are into the developing towards the complete proteome atlas and tissue specific proteome atlas of pigeon pea. So For this, first we have standardized the total protein extraction protocol, and then perform the 2D and perform all sort of bioinformatics analysis. And during this analysis, we identify that DOF is the common protein which is found in all the stress condition also, like salinity and drought. And also this studies confirm that other other legumes and cereal crops, let's say in the soybean, arabidopsis rice, they are having the DOF. So similarly, in case of pigeon pea, we found this DOF domain which is having the crosstalk between different kinds of abiotic stresses, right? Now my group is also focusing on the machine learning algorithm. And this is work of my postdoctoral research actually, where we develop this cassava, because cassava is also one of the most staple crops, especially for the African countries, and try to map the development of third generation haplotype map, and also develop the cassava assembly software. So that generally we used to have the uniform assembly, like we don't go for the hybrid. But here what we have done, we have used both long read and short read, and go for the hybrid assembly, and develop one software gas pipe, and with where we can perform the just simply send your all the fast queue files, and it will and do all the analysis, and finally give you the sequence with in the form of haplotype purge. And this is the pipeline we followed, and also make some of the fine tuning in this model. So we apply some of the machine learning model based on some of the features. So this is actually machine learning model, supervised machine learning model, where we have the some our training data set, out of which we tested, and based on some of the decision tree algorithm, and try to map and identify the true variants. Because getting out of this like millions of snips, if I go back, and you can see that here I am having like 32 million snips, right? So out of 32 million snip, how I can identify that which are the true variant, because we can't go with the all 32 millions of snips. So this is the file, the haplotype call file, and you can see the cassava assembly starts with the different species and different genotypes, and the length is also very quite, and the N50 and their length. And this is the statistics of the hybrid assembly, which is based on the haplotype information, because haplotype is in which we can have the gene, all the gene are sorted in a same array. And other than that, I was also working in collaboration with identifying some of the this cassava mosaic virus. And we have target cassava mosaic virus. For this, we have target RAP, replication associated protein. And we have collected the protein from different strains of different strains across different countries, like Nigeria, India, Eastern Africa, South Africa, and estimated perform all sort of the primary, secondary, and tertiary analysis, and identify some of the common pockets across all the different countries. And we perform the protein ligand docking interaction, and identify some of the key things. So in my takeaway, I would like to say that now it's not a time to stick on one particular omics area. We need to have the multi-omics approaches, so that via validating all those omics, we can have we can able to say that yes, this is the key thing which identify. Because some of the things which really works in genomics, but if we go for the protein, so because post translation modification happens and something like that, so we need to check with the different omics, and then go with that. And now the arena is for the machine learning and deep learning, so we can have a use data. For example, in case of protein structure prediction, we are having AlphaFold now. So we can have we can input lot of sequence, and we can mine the structure. So that way we can do something in case of genomics, so it will be great boost. And so that my takeaway is the integration of multi-omics approaches can be utilized to boost the genetic gains in the crops. So these are the some awards and accolades which I received for my work. And this is the some interesting thing which I found during my postdoc, that hackathon, in which we are having 3 to 4 days of hackathon, which in which we try to develop some of the codes and model, and address some of the things. So the cassava hapmap 3 we could able to do within, like, short of 1 or 2 week, because of this hackathon. And yes, thank you all for your patient listening, and the funding agency, and my yeah, supervisors, both PhD, postdoc, and my group also. So my last word is we need to envision our research goal beyond 5 to 10 years, concern, demand, and problems, so that we have a sustainable research goal also as the sustainable development goal, and it should be lies with the both the way. Thank you so much for your patient listening. With this, I will uh, close my presentation, and I will be happy if you have any question. Sakshi.
Thank you so much, ma'am. It was very well explained and knowledgeable session. So it's time to take some questions quickly. We have some
Yeah, yeah,
questions.
yeah, I can.
Yes, why SNPs are important in NGS?
Okay, so okay, as we all know that the single nucleotide polymorphisms are the most abundant DNA marker in the plant genomes, and every individual is 99.9% similar. 0.1% variation is because of the SNPs. So that's how people earlier, if you see about the marker history, people used to have the RFLP, then RAPT marker, then SSR, microsatellite, then they rely on and stick on the SNPs because they are highly reproducible. You and the data analysis is also quite easy, and we can, but the key thing here is which snips you have to taken into account. For that you need to have a good bioinformatics algorithm based on some key score and like p convert score or some of the p value, where we can see say that yes, this is the snips which is considered to be as a true variant, and we go further for the mapping or using this particular for our study. So that's how the snips is important. And also now you can see the example of personalized medicine. It's all because of the SNPs, because some of the drugs is may work on me but not in my mother or my husband. So that's the thing, because their genotype is entirely different, maybe.
Correct, correct. Yes. Next question. How to save germ plasma of rice?
Yeah, so in case of germplasm, like we have in NBPGR, National Bureau of Plant Genetic Resources at IRI New Delhi, like we have collected, so we have deposited first our germplasm, and then a few we can ask them, and then we can multiply, and then also you can store like in a 4 degree or something like that, or we need to revive. So that's how we can save our germplasm. So for that, like in case of animal also, we have NBAGR. So they are the repository where we can store our the germplasms. Like GenBanks is there.
Okay, okay.
So it's
Next
not
question.
implied for the rice; it's for the other crops also.
Any tool suggested for whole genome comparison?
So for the whole genome comparisons, like it's depend upon how what you want to compare. If it is like the base pair by base pair comparison, then Minimap 2 is there. It's for the genome level.
Okay, okay.
Or if you want to
I
go for the Synteny analysis, then the SynMap is there. SynFinder.
okay, okay. Uh, Thank you, ma'am. I think we have done with all the questions. So thank you once again for answering all
Okay,
the
one
questions.
question is Ekta Singh is saying that congratulations for the wonderful session. My question is not scientific. How do you
Okay.
justify that modification of genes and going
And
against nature? Okay, wonderful questions. But yes, it is scientific, I would say. If you I would say that if you don't want anything and you want edit, it means it's not required anymore. And if it is required, so you can, what you can do that first you let's say if you're having 10 seeds of that particular germplasm and you won't go for the editing, so you just keep 5 seeds for your as a reservoir. You don't disturb them, and in the 5 you can go for the editing. Because yes, it's not like that it's a washing that you can redo the things. Once you have edited, it's for permanent, right? So but if you are editing something for some good cause, it means you don't require anymore that thing. No, that's why we are opting that. Otherwise, why?
yes, correct. Well said, ma'am. So do we have any more questions? Okay. Okay, I think we have done, ma'am. So it's time to wrap up. And I would say thank you so much once again for answering all the questions and for such a great presentation. It was wonderful to have you with us. And of course, it won't come to the end without mentioning it proudly that this Women's Day Special campaign initiative by the Altum Technologies to acknowledge the effort of the women researchers in their respective sectors is an appreciated step. So for thank you once again to all of you. For any query, you can reach us at altum.com. We are available on LinkedIn, Instagram, Facebook, and on every social media basically. So like us, share us, subscribe us to get the updates in the field of life sciences and many more. We always work to deliver insightful and cutting edge content in the drug discovery and the development. Thank you all. Thank you for joining us in this campaign. Thank you. Thank you so much.
Thank you,
Thank
Sakshi,
you,
and
ma'am.
thank you all for having me and listening me. Have a
Thank you,
great
ma'am.
Holi to you all, and happy Women's Day.
Happy Holi. Yes, happy Women's Day, ma'am. Thank you.
So I log out, Sakshi, na, now.
Yes, ma'am. Yeah.